The overall goal of this protocol is to observe the development of Tribolium castaneum embryos non-invasively over very long periods of time.
Light sheet-based fluorescence microscopy helps to answer morphogenesis-related questions in the emerging insect model organism Tribolium castaneum, addressing cell migration and proliferation patterns from the early blastoderm to the onset of muscular movement.
The main advantage of this method is that critical factors such as photobleaching and phototoxicity are kept to a minimum even when the embryos are observed for several days.
To mount an embryo into the sample chamber using an agarose column, first fill a 1 ml syringe with distilled water and use a small rubber hose to attach the syringe to a glass capillary. Fill the capillary halfway with distilled water. Then use a paintbrush to transfer an embryo to the inner side of the glass capillary opening and stick the capillary into the liquid agarose, slowly drawing a few microliters of the agarose along with the embryo into the capillary. When the embryo is in place, there should be a few microliters of agarose both above and below the embryo. When the agarose has solidified, shorten the capillary to a length of about 5 mm. The remaining fragment should be completely filled with agarose, and the embryo should be found within the second quartile. Then insert the steel cylinder with the pin into the sample chamber and stick the capillary fragment on top of the pin. The agarose column should protrude a few millimeters from the region of the capillary fragment that contains the embryo.